Hey everybody, Professor Powers here. Here's chapter 3 lecture, which is the second chapter in our first module, and getting through this week. So without further ado, let's move on to the first slide. All right, the cell theory helps us understand where cells come from. So we know that the basic unit of life you learned in chapter 1 is the cell. We do have two different kinds of cells, the prokaryotic cells and eukaryotic cells. So it doesn't matter what kind of cell you have. If you have a cell, you are considered alive. Now we know that all living things are made up of cells, so that makes sense and lends support for the first bullet there. And then lastly, new cells arise only from pre-existing cells. So when we think about mitosis and meiosis and how are we replicating ourselves and making new ones, well, it really ties back into the cell theory. So we'll be getting into that, I believe, next week. So one of the themes that you're going to learn is that structure follows function, okay? So it's no different when we're taking a look at maybe an organ or a particular part of your body, or when we're taking a look at cells. Cells vary in their structure and function. So red blood cells, for example, are kind of unique because they have a concave center. It's almost like somebody pinched that red blood cell. So at maturity, our red blood cells lose the nucleus, and the reason it loses the nucleus is so that the hemoglobin protein can carry iron, okay? So cell structure and function. The cell's function is being dictated by its structure, okay? Take a look at nerve cells. You've probably taken a class before, likely psychology, and that central star here is the cell body of the neuron, which is a nerve cell, and then you have short extensions that are the dendrites, and you have long extensions, which are the axons. And this is how one neuron communicates with the next. This is how we send electrical impulses. This is how we are going to determine which neurotransmitters and how many neurotransmitters are going to be released at the synapse. Okay, so all the features of this nerve cell are going to essentially allow it to do its function, which is just to rapidly send messages. Now, I love bone. I love the slide of bone. Bone shows us that it's very, very, very rigid. Now, it doesn't take a lot to realize. You can pound your humerus and feel that there's this super rigid structure inside. And what it is made up of are little strands of connecting fibers, if you will, surrounding what we call the bone cell, an osteocyte. Okay, so essentially what we see is that the bone is highly reinforced, and the cell itself is designed to be highly reinforced with the other cells around it. So we get something that looks like almost like a tree trunk, which is making up our bones. So it's these repeating tree trunks, which are called osteons, making your bone. So it's really a pretty fantastic area of biology. All right, so you're going to have a chance to work with a microscope in your virtual labs, but a couple of things I wanted you to think about. The first one is resolution, which is the ability to distinguish between adjacent points of an image, okay? So different kinds of microscopes work differently. And so your ability to distinguish between 2 points gets better and better and better when you use like the really super fancy microscopes. You're going to use a light microscope in virtual labs, and then I'm going to show you some transmission electron micrographs, which will give you an idea if we bombard the specimen that you're looking at, maybe it's bone, with electrons, how that radically changes what we see. So we know that microscopy has aided our ability to see. Its technology is aiding our ability to learn more about science, okay, which is pretty exciting. Okay, so what we see here is essentially the view through a microscope of human red blood cells. On the far left, this is your normal light microscope, so one you would use in a virtual lab, one that you would use on campus in our labs. The second one is what we call transmission electron micrograph. So it's going to kind of make things pop, and you're going to see some materials like here. We're going to see the vessel. So this is likely a vein. It could be an artery. So you can get some more detail there. Certainly we see that in this red blood cell, we actually have some sickle shaped. So this person probably has sickle cell anemia. But look over here, the scanning electron micrograph. now gives us like this 3D image, okay? And so microscopy is going to aid our ability to see. And this aids our ability to study and to learn and understand how structure follows function. Okay, so this should be a review for you, so I don't want to spend much time on it. If you need to pause my video, you can. If I talk too slow, you can speed up my videos. If I talk too fast, you can slow down my videos. Here we go. All right, so when we take a look at cells, there are 3 features of all cells, whether they're prokaryotic or eukaryotic. There's the outside membrane, plasma membrane. It surrounds the cell. We're going to see that it's made of what we call phospholipids. So lipids, think oil. This phospholipid layer, there's 2 layers, so it's called a bilayer, one on the outside of the cell, one directly inside the cell before you get into the cytoplasm and all the structures inside. This lipid bilayer is going to have to regulate what can come in and what can go out. You know, it's not just like an open door all the time. Come on in, come on in. It's not like that. Instead, it is going to be selectively permeable. So it's going to differentiate what's going to come in and out. We'll come back to that. The cytoplasm is this semi-fluid, so think thick nutritional broth inside the cell, pushing out on the cytoplasm. And, the cytoplasm is this thick nutritional broth pushing out on the plasma membrane. The cytoplasm is going to have structures floating in it. If I am a prokaryotic cell, I'm in the kingdom Archaea, I'm going to essentially have free DNA that's going to be suspended in that cytoplasm. And I'm going to have some ribosomes. Cool. If I'm a eukaryotic cell, much like algae or plants, then I'm going to have a nucleus, and I'm going to have endoplasmic reticulum, and I'm going to have a chloroplast. These are my organelles. And essentially what we're going to have is all these structures, again, floating around inside the cytoplasm. So just remember, differentiate between prokaryotic and eukaryotic cells. So this is one of these scanning transmission micrographs using that particular kind of microscope that allows you to really start to see structures inside cells. You don't see this in our typical lab microscopes. So I thought that was pretty fascinating and interesting. You can come back and study that if you'd like. Here are many of the features. I didn't put every single one of them on the study guide, but many of the features that are typical of a eukaryotic cell, and this is an animal cell. We're going to spend some time in the rest of this video going through the structures inside the cell. Okay. When we take a look back into evolutionary history, what we see is that the first cells on Earth were prokaryotes, the archaea and the bacteria. So there's some interesting things that happened in the evolution of those cells to help them survive in those environments. And when we were able to do DNA technology, we discovered DNA technology, and now we have some pretty fantastic ways to study evolution through DNA technology, we can see that the eukaryotic cells are more closely related to the archaea than we are the bacteria. So archaea include things that grow on like hot springs and thermal vents on the bottom of the floor and like bogs where there's a high methane environment. Our cells are more closely similar to them. So one thing that people will think about, like when you're thinking about the evolution of a eukaryotic cell, prokaryotic cells didn't have a nucleus, didn't have endoplasmic reticulum, Golgi, chloroplast, mitochondria. But what we know is that those structures, particularly good evidence with mitosis and, did I say mitosis? Good evidence with chloroplasts and mitochondria is that they are actually endosymbiotic bacteria living inside of the cell. And so they resulted from what we call endosymbiosis. I'll explain that a little bit deeper later. This shows you some figures. Again, I'll come back to this because the big word here is endosymbiosis, but also it's going to involve what we call phagocytosis, where one cell, this bigger cell, consumes and brings inside of its cell through bulk transport another cell. Okay, so let's take a look. Plasma membrane, you already know a little bit about it. It is going to be selective. So there's a couple of things to know here. The fluid mosaic model means proteins freely move laterally and contains cholesterol for support. So part of that cell membrane or plasma membrane is going to be fluid mosaic, okay? And then part of that membrane is going to be made up of glycoproteins and glycolipids, which essentially when you see glyco, think carbohydrate. These carbohydrate chains are attached to proteins and lipids, and essentially what they're going to do is they're going to let the cell know what belongs to it and what does not belong to it. So those glycoproteins and glycolipids can act as receptors, starting to select what comes in and out. And here's a lovely figure. The lipid bilayer here looks kind of funny, but essentially it's a phospholipid layer, and there's two of them. What you have are the lipid tails to the inside because lipids don't like to mix with water. So out here you have your phosphate head. This is showing us outside the cell and inside the cell. And then you get an idea of these glycoproteins, here and there, glycolipids. And we actually have an integral protein here, peripheral protein, which are also going to help select for things moving through or into and out of the cell. You can pause and read more about this. I'm going to move on. Selectively permeable, meaning some things can come in and some things cannot come in. We are going to talk more about this when we talk about active transport and passive transport. So passive transport, no energy is required, and you're going to have a little more in-depth work with this in your lab, your virtual lab. Diffusion, osmosis, and facilitated diffusion. So essentially, these are examples of passive transport. Essentially, what you have is a building up of a concentration gradient that is going to easily move particles or move substances from regions of high concentration to low concentration. Active transport involves bulk transport. So remember when I said you have one cell, I'm thinking about an amoeba. An amoeba eats bacteria and yeast. I've seen it happen. And they take out their fake feet or their pseudopods and they surround the thing that they want to eat and they bring it across the plasma membrane. Now you have a plasma membrane surrounding the very thing that they're trying to eat. Living things. Okay, if I'm consuming a bacterial cell, you know, it's got essentially all the parts. So essentially you're consuming its cell wall and its own cytoplasm and the stuff inside the cell. Okay, so endocytosis. Exocytosis is when you want to push things out. So again, amoeba can get rid of waste products from what we call contractile vacuoles by exocytosis. Think about it as the cell just dumping out all the waste products it doesn't need. Okay, so active transport is different than passive. Passive transport does not need energy. Active transport needs energy. And a little bit more complex process. Okay, so in the next couple of minutes in this video, let's talk about diffusion. So if I have a candle here, if I went and burn this candle, the thing is it's going to smell up my room. It's going to smell so good. It's going to smell like coconut pina colada. And then over time, the high concentration of those scent molecules essentially are going to move from this little room in here out into the rest of the house. So it might take 10 minutes, 15 minutes before my husband goes, oh, did you burn that candle again? Yeah, he doesn't really like scenty things. So it takes a little bit of time for it to get there. But essentially what happens is this random movement of molecules, and they're going to keep moving and keep moving and keep moving until they are equally distributed. Now, another example is if you take like food coloring, and you have some water and you put 1 drop of food coloring in the water and you just sit there, you can see it, the blue drop is dropping down, 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 down in the water, and you give it some time, you come back, that blue dye will have moved from the region of high concentration to low concentration because of this random movement, and then the whole, cup of water would be blue. Okay, so in this case, molecules are doing it passively, there's no energy required, and the molecules are moving in both directions. That way you can get that equal distribution. I will need to make a second video to finish up chapter 3, so I will pause here and I'll see you in a moment.